Hello everyone. Hello all the students who are preparing for CSIR UGC NET. This is Tithi here, and today I am going to discuss some of the important Unit 8 previous year questions. Now the topic that I will be covering today will be on mutation and complementation test. Now these are two topics from where a lot of questions come. Complementation test related questions did not used to come some 8 or 6 years ago, but nowadays, like in the couple of sessions what I have seen past sessions, every time there is a question coming from complementation test. So definitely it has become one of the important section from where questions do come. And if you once you understand and once you find out understand how to recognize a question from complementation test, the question becomes very easy to solve. So let's get started. We'll start with the question on mutation. Now in mutation topic, this type of question is very common. Now in this question, what they give? So they give some unknown compound or a new compound, and that causes a kind of mutation. And to determine what kind of mutation it is causing, so scientists will use a known mutagen to do the reversal of the mutation. Okay, so this entire study is called as mutation reversal study. Mutation reversal study. Okay, so lot of questions have come from mutation reversal study, and this is a topic which has remained a favorite of all the question setters, and almost every session, or like every year, okay, every alternate session, for sure one question of this type comes. Now this type of question, it's very easy to solve this type of question. The only thing you have to remember are the different mutations and the different mutagens which can cause those mutations. Okay, so mutagens and the mutation that they cause. If you remember this too, it's very easy to answer this kind of question. Now let's see this question. So two new chemical compounds X and Y are synthesized in a laboratory and tested for their potency as a mutagen. The nature of the mutation produced by these compounds is tested for reversal by other known mutagen, and the following results were obtained. Which statement best describes the nature of these two mutagens? Okay, so this mutation reversal study is given, so first thing you have to determine is what all known mutagens are used for reversal and what will be each of their function. So 5-bromouracil, what kind of mutation it causes? It causes transition base substitution. Okay. EMS stands for ethyl methane sulfonate, which is an alkylating agent, and that also results into transition. Hydroxylamine also causes, it's a hydroxylating agent and it also causes transition. Okay. Acridine orange. Now acridine orange is an intercalating agent. An intercalating agent, they intercalate between the adjacent nucleotides, and that results into insertion or deletion mutations. Okay, now deletion is, or insertion or deletion, this is not of a long stretch of nucleotide but single nucleotide. Okay, so insertion or deletion of single nucleotide. Okay, now once you have determined this, now compound X, how it is getting reversed? So it is getting reversed by 5-bromouracil and EMS. Now both of them causes transition. Okay, so if the transition is reversing the mutation, means the original mutation was also a transition, right? So X is causing transition. Now, if you're thinking that if it is causing transition, then why hydroxylamine is not able to reverse it? That is because hydroxylamine cannot do both the side transition. Okay, so it can both the side transition means see AT to GC. It is a transition, right, where a purine is replacing a purine, pyrimidine is replacing a pyrimidine. Now similarly, CG to TA will be the reverse transition. So fibromouracil, EMS, they are all able to do transition in both the direction, but hydroxylamine can only do in one direction. Okay, so because the reversal needed a different direction in which compared to the one in which hydroxylamine can do, so that's why transition is not caused, the reversal is not caused by hydroxylamine. Okay, for this experiment. Next, for compound Y, now compound Y is not getting reversed by any of them. That means for sure Y is not a transition. Now it is getting reversed by acridine orange. Acridine orange, it causes insertion or deletion, mutation of single nucleotide. So if the new compound is acridine orange, Y is getting reversed by acridine orange, means Y is causing insertion or deletions. Okay, so compound X produces single base substitution that generates CG to TA transition, and compound Y produces insertion or deletion. Okay, so our answer is option A. All other options, if you see, so X is saying insertion or deletion, not possible. X produces single base substitution and Y also produces transition, not possible. Compound X produces single base substitutions and generate insertion, Okay, so X is causing insertion according to this D, so that is not true, and Y is producing deletion, not true. Okay, so answer is option 1. Now this type of question you will get many varieties. Most of the time they will increase this number of compounds here. Okay, but the ultimate way of how to approach this question remains same. You have to know the mutagens, you have to know what kind of mutation they cause, and you have to correlate it with the new compound or the action of the new compound. Another question which is very frequently asked from this topic of mutation. So mostly it comes from the from this chromosomal aberration. Okay, so it is like a diagram like this. If you have gone through the previous year question paper, you will see this kind of question very common. Okay, now what is this question is saying? An individual is having an inversion in heterozygous condition. The regions on normal chromosome are marked as capital A, B, C, D, E, F, G, while the chromosome having inversion has the regions small a, B, C, D, E, F, G. The diagram given below shows pairing of these two homologous chromosomes during meiosis, and the site of crossing over is indicated. Okay, so this X is the site of crossing over. The following statements are given to describe the inversion and the consequence of crossing over shown in the above diagram. Now, from which topic this is coming? So this, as I told you, comes from chromosomal aberration. Now, inversion is a type of chromosomal aberration or chromosomal mutation in which a segment of the chromosome is inverted. Okay. Now, if inversion happens in heterozygous condition, heterozygous condition means only one of the homologous chromosome has the inversion, whereas the other member of the homologous pair, the arrangement of gene is same. Okay. Now, if that is the scenario, then definitely the chromosomes, the two homologous chromosomes cannot align with each other at the time of crossing over. Now, for crossing over to proceed, that is a basic necessity. So, to match up or to align, what it will do, so the chromosome which has the inversion, it will create a cross loop like this so that alignment can happen. Okay. Now, because this aligning is a trouble, and also when the crossing over happens and the chiasmata resolves, okay, so there are the recombinants that will be produced will be inviable. Okay. Now, what will be the exact consequence of crossing over occurring through the heterozygously inverted segment? That will depend on whether the inversion is pericentric or paracentric. Okay, so here the consequences are written. But to answer this question, the first thing that you have to do is you have to find out whether the inversion is pericentric or paracentric. Okay, now what is pericentric and paracentric? So pericentric inversions are those inversions which include centromere. include centromere. Okay, so the inverted segment will include centromere. Paracentric inversions, they do not include centromere. Okay, so now we have to find this out, whether, okay, just writing centromere. So here now we just have to find it out whether this particular inversion is pericentric or paracentric. So for that, how will you find it? So I told you that cross loop will be formed. Okay. So whatever genes are coming within that loop is the one which has got inverted. Okay. Now you see it is clearly can be seen that these black dots are the centromere. Okay. Now are these black dots coming within centromere? So are these black dots coming within the loop? No, they are outside the loop, right? So within the loop, which all gene segments are there? C, D and E. Means this is the segment which has got inverted and it does not include centromere. So if it does not include centromere, then what kind of inversion it is? Paracentric inversion. Okay. So first of all, so it is paracentric inversion. So this option F is correct. Okay, and immediately A becomes wrong. Now, what will be the consequence of crossing over occurring through the heterozygous paracentric inverted segment? So, the consequence will be that when this chiasmata has to be resolved, it will result in the formation of a dicentric chromatid and an acentric chromatid. Okay, that acentric chromatid will be lost. Okay, because it will fail to segregate as centromere is not present, and the dicentric chromatid, it will be pulled to two different poles, which will create a dicentric bridge, which will make the chromatid to abruptly break from the middle. Okay, so as a result, what will happen that the recombinant, so there will be 4 gametes formed as after meiosis. So the gametes which are receiving the chromatids that participated in crossing over, the recombinant chromatids, they will be inviable. Why? Because of deletion of some genes. And the gametes which are receiving the chromatids which did not participate in the crossing over, because in crossing over only 2 of the chromatids will participate, right? Other 2 will not, out in a tetrad. Okay, so the chromatids that did not participate in crossing over, the gametes which will receive those chromatids, they will be absolutely viable. Okay, so it is a paracentric inversion, that is correct. Now B says that it will generate dicentric and acentric chromosome following separation. That is also true. C is wrong because it will not generate monocentric recombinant chromosome. D says that all the gametes thus formed will have deletion or duplication and will be non-viable. That is also wrong. Why? Because 50% of the gametes which receives the chromatid that did not participate in crossing over, they will be viable. Only the gametes that receive the recombinant chromatids, they will be non-viable. So option E, which says that 50% of the gamete having recombinant chromatid will be non-viable, where other 50% will be having non-recombinant chromatids, will survive. Okay, so answer will be option 3, B, E and F. Okay, so like this question can also come for pericentric inversion. So you have to shift the consequence of that. So first of all, now that inversion, whether it is paracentric or pericentric, might be mentioned to you in the question, but usually it won't. And looking at the image, you have to determine whether it is paracentric or pericentric. Okay, so how will you find it out? Look in the loop and see whether in the loop centromere is there or not. If it is not there, like this condition, it is paracentric. If it is there, then it will be pericentric, and accordingly the consequence of crossing over occurring through the heterozygous inverted segment will change. Okay, so these two were the very commonly asked questions from mutation. Now moving on to complementation test. Okay. Now complementation test, what is it? So it's a test, it's a crossing based test which is done to find out whether the mutants, two different mutants, whether they have two different mutants which are having the same mutant phenotype, whether they have the mutation in the same gene or different gene, right? So that is what we try to find out in complementation test. Now the question which comes from complementation test, it will never be mentioned anywhere that whether it is in the exact words that we are doing a complementation test. No, you have to find it out. How? So the key will be to see whether two or more mutants are getting crossed or not, and then we are seeing to find out whether it is producing the wild type phenotype or mutant phenotype. Okay, because that is the theme of complementation test, that we cross. So what exactly is done in complementation test? Is we will cross two mutants. Okay, mutant 1 crossed with mutant 2. Now, which are those two mutants? The mutants which have the same mutant phenotype. Okay. Now, it can have two different results. So, if mutant 1 cross mutant 2 produces mutant phenotype. Okay. That means, what will be the understanding? That the genes, these mutations, they did not complement each other. Why they did not complement each other? Because the mutations were occurring in the same gene. Okay. Now, if mutations occur in the same gene, means it is producing alternate form of the gene, which we also call as alleles. So, this mutation will be called as allelic mutation. Okay. And if allelic mutations are there, that means no complementation. Now, if mutant 1 cross with mutant 2, we get the next generation becomes wild type, okay, which is having the wild type phenotype, okay. So, you're crossing two mutants, but the progeny is having wild type phenotype. How is it possible? It is possible when mutant 1 and mutation 1 and mutation 2 are complementing each other. Now, how can they complement each other? They can complement when the mutations are in different genes. And if they are in different genes, then they will be non-allelic mutations, because they cannot create alleles of each other anymore. So, they are non-allelic mutations. Okay. So, all the questions based on complementation test can be answered by this simple understanding. That if mutant 1 is crossed with mutant 2, if the resultant progeny is also mutant, means no complementation happened, means the mutations were in the same gene, which makes the mutation allelic mutation. If mutant 1 crossed with mutant 2, we get wild type phenotype, means complementation happened, means different genes are involved here, and non-allelic mutation. Okay, now let's get back to our question. So, this was a question which was asked in this December exam, okay, and like couple of years back, a couple of years previous paper also, if you see, every session there has been a question like this, okay, related to complementation test. So, what is this question saying? P. aeruginosa makes a blue-green pigment called as pyocyanin. To understand the pyocyanin biosynthetic pathway, mutants which cannot make pyocyanin were isolated. Okay. So, see, all the mutants have same phenotype. Why? What? That they cannot make pyocyanin. 6 such mutants are crossed. So, we are crossing mutants, okay, with each other, and the data is given below. I told you to find out whether it's a complementation test or not. This will be the clue, that you cross that in the experiment you are crossing two mutants having similar phenotype and then checking the result. Okay. Now, minus means pyocyanin negative, means a mutant phenotype. Pyocyanin negative is a mutant phenotype. Okay. Pyocyanin positive is a wild type phenotype. Okay. Now, based on this data, can you predict how many genes are responsible for pyocyanin production? Okay. So, for this, what we have to find out, how many complementation groups are there. Okay. So, whatever is the number of complementation group, that many genes are involved here for the pyocyanin production. So, as you can see here, mutant 1. So, mutant 1 is not complementing itself. Definitely it will not, because it is the same mutation. Along with that, mutant 1 is also not complementing 6. Okay. What does that mean? That 1 and 6 are present in one complementation group. That's why they are not complementing each other. Okay. Now moving on with 2. Now 2 is not complementing only itself. It is complementing all others. Okay. That means mutation 2 is in a separate group compared to all other mutants. Same with mutant 3 and with mutant 4. They are not complementing only themselves. And with mutant 5. Okay, they are complementing all other mutations. So whatever mutations they are complementing means they are different from them. Okay, and so that's why they all are in 1 gene. Okay, 6 we have already placed in the group of 1 because 6 and 1 are not complementing each other. Okay, so these are the different complementation groups. In terms of genes, what they are all different genes. So how many different genes are there? 1, 2, 3, 4, 5. Okay, so even though like we have taken 6 different mutants, but mutant 1 and mutant 6 were having mutation in the same gene. Okay, so total number of genes which are involved here for pyacinin production is 5. I hope all of you understood this. Okay, now let's take another example. Now this was also asked a couple of years back in CSIR, just couple of years, not later than that. So a Drosophila mutant line A with vestigial wing is isolated in a laboratory. The vestigial wing phenotype was observed to be recessive and mapped to gene X. 3 other laboratories also isolated vestigial mutants called line B, C and D. In order to test if the mutation in line B to D is also mapped to gene X, the following crosses were made and phenotype of F1 progeny observed. Okay, see again what we are doing. So vestigial wing is the mutant phenotype. So mutants A, B, C, D having the same mutation are getting crossed and the phenotype of the F1 generation is noted. So based on the above, identify the lines which is most not likely not to have the mutation in gene X. Okay, so in the same way we will do. So vestigial means mutant phenotype, normal means wild type phenotype, right? So what do we see? A cross B is mutant, means A and B are in the same group. A and C is also mutant, means C is also there in the same group and that is also satisfied. So AB is mutant, BC is mutant, and when you cross AC, so BC also you see is mutant, right? And when you are crossing CA or AC, that is same, so that also is mutant. Okay, so A, B, C are in the same gene. So it is the same gene that has A, B and C. Now what we have done here that so gene A, this mutant line A is already crossed. The mutation is already mapped to gene X. Okay, so since A, B and C are happening in the same gene, that means they are also happening in which gene? Gene X. Okay, now what about D? See here, A and D is normal. So you cross 2 mutants and you are getting normal phenotype. Similarly, B and D normal, C and D normal. That means D is in a different gene. It is not present in gene X. Okay, so what is the question? Based on above, identify the line which is likely not to have a mutation in gene X. So what line will it be? That will be line D only. So answer here will be option C. Okay, so you can see that the procedure is same. The same technique you have to perform to find out the different complementation groups or different genes that are involved here, and accordingly you have to answer. Okay, so here how did we do? So we found out that D is not in the gene X. Okay, so that's why D is in a different gene. So D cannot be on gene X. That was the question. Okay. So there are more questions like that, and I want you all to practice. And of course, I'll be coming back again with more question discussions on Unit 8 topics. Thank you, everyone. Bye-bye. Have a great day.